In this video, we will discuss the concepts and applications of fluorescence spectroscopy. Topics covered will include basic background theory, how it works, and some instrumentation. There will be a special focus on applying fluorescence to monitor peptide cleavage and enzyme activity. So to begin, what is fluorescence? Fluorescence is a member of the luminescence family, categorized as one of the two types of photoluminescence. Specifically, fluorescence is the property that some atoms have that enables them to absorb light or electromagnetic radiation at a particular wavelength and then subsequently emit light at a longer wavelength after a brief time interval. The time interval is known as the fluorescence lifetime. So then, what constitutes a molecule's ability to be fluorescent? In order for a molecule to fluoresce, it must contain a fluorescent probe, a fluorochrome, or a simple dye. These are otherwise known as fluorophores. The definition of a fluorophore is chemical compounds that have the ability to re-emit light upon excitation. Fluorophores can be intrinsic or extrinsic. Intrinsic fluorophores are naturally occurring. Examples of intrinsic fluorophores are aromatic amino acids or neurotransmitters. Extrinsic fluorophores are synthetic dyes or modified biochemicals that can be added to a molecular compound. Fluorophores can be conjugated with larger macromolecules like enzymes, proteins, or lipids, so they can be analyzed using fluorescence spectroscopy. A molecule's ability to fluoresce is a process of excitation and emission that occurs in 3 steps. We'll use a Jablonski energy diagram to walk through the process. The diagram is a representative figure of the electron activity during the excitation and emission steps. The first step is the excitation of a molecule by an incoming photon. This means the electrons are excited from the ground state to higher excited energy states. The excited states are dependent on each particular molecule. Similarly, each fluorophore has a specific excitation wavelength that can be measured. The second step is the vibrational relaxation of the excited state electron to the lowest energy level. The third step is the emission of the photon at a longer wavelength than it was absorbed. This returns the molecule or the compound back to the ground state. Like the excited state, each fluorophore has a specific emission wavelength. All 3 steps occur in a billionth of a second, with the fluorescence actually occurring between about 10 to the negative 10 to 10 to the negative 7 seconds. There are several reasons as to why molecules don't fluoresce even in the presence of a fluorophore. When a molecule contains a fluorophore, the blue circle, but no fluorescence is observed, this is due to quenching. This is done by a quenching agent, the green circle. Quenching decreases or eliminates the fluorescence intensity of a molecule by the transfer of energy to the quenching agent. The 3 major forms of quenching are complex formation, collisional, and energy transfer. Additionally, solvents and other environmental factors can affect fluorescence emission. Solvent composition and factors such as pH, temperature, and localized concentrations can impact the ability of a molecule to fluoresce or its intensity. This happens because of dipole interactions and solvent relaxation. Let's look at an example of how fluorescence can be applied. In this example, we will look at a protein that is degraded via protein cleavage by an enzyme and how the quenching agent and the fluorophore of the protein interact with one another. The quenching mechanism is a long-range resonance energy transfer mechanism, meaning that the quencher absorbs the energy from the emission stage of the electron excitation, causing the molecule to not fluoresce. The protein will be represented by a fluorescence resonance energy transfer threatened peptide with a generic enzyme. Shown is the threatened peptide sequence. For simplicity, let's refer to it as protide. The X's represent the sequence of amino acids of the peptide. The left side is a nitrotyrosine, which acts as the quencher, and the right side is an anthrolamide lysine, which acts as the fluorescent donor or the fluorophore. When the peptide is intact, there is no fluorescence because the quenching agent is absorbing all the energy from the emission of the fluorophore. Let's say the peptide is cleaved between X4 and X5 by the enzyme. This cleavage would separate the fluorophore end of the peptide from the quencher end. When the quencher is no longer in close proximity to the fluorophore, it can no longer absorb the energy given off by admittance. As a result, the fluorophore is able to emit fluorescence. As more of the peptide becomes cleaved, the fluorescent intensity increases because the concentration of unquenched fluorophore increases, emitting off more fluorescent light. Because of the broad range of studies that fluorescence can allow, fluorescence spectroscopy is widely used. It is becoming an important tool in genetics and cell biology because of sensitivity and the high specificity fluorescence requires. Additionally, intracellular dynamics such as enzyme reaction rates, binding constants, and reaction mechanisms can be studied using fluorescence. Thanks for tuning in, and may the fluorescence be ever in your favor.